Already started, I think. Yeah.
Okay.
User friendly. Seems I know.
You can see my screen now? Hello? You are able to see my screen?
Yes.
There was a center problem.
Okay. There, at your end? See, Osama and Kimia, whenever I'm asking a question, you can just raise your thumb like this. Okay, if it is okay, and if it is not okay, then you can put your thumb down. So then I'll understand. So now you can see my screen.
All right. Okay.
Okay, okay. Okay, thank you, thank you, thank you. Yeah, Osama and Kimia, for the entire group, I'll just tell you how we have planned the entire training program. Basically, if you remember, we had a similar training program, I think in 2021, right? So where I had done basics of oncology and then we had moved to products. But now we are short of time. So taking basics of oncology will be really difficult. So I cannot take basics of oncology because that takes almost 1 day, and then we have almost 15 products to complete. So what we have done basically this time is we are taking some of the selected topics of basics of oncology, which will actually help you in understanding the products. Okay. So basically we'll be touching on the main cell cycle and the classification of all the chemotherapeutic drugs and what is the mode of action of each of the drug. So that will make your understanding with respect to products easier. Okay, so I have just taken two topics in basics of oncology, and then we will move to the products. So once you understand the chemotherapeutic drugs and their mode of action, then understanding products will be very, very easy. And then when we go to the products, what I'm going to do is I'm going to basically give you the classification of the drug, how it acts, and which are the indications, and which are the different doses which are used. Okay, and the major adverse effects. So we will stick to only how much is relevant, right? And later on, after this training program, we can plan a detailed training session whenever it is feasible for both of us. So that will be a very detailed training program. Okay. So this is how we'll be going about. So Kimia and Osama, that's fine. We can start now. Yeah. Okay, 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 okay. And I'm sure a couple of you have already attended my training program, so you are familiar with some of the basics, right? With respect to oncology.
Yes.
Great.
Ma'am, you are muted. Ma'am,
Mr. You have, I think, mute.
you have to unmute yourself. Yeah.
Yeah. So we all know we are going to discuss about cancer products. So we are in the field of oncology, right? So oncology is nothing but the field or the science which deals with cancer, the treatment, and all the aspects related to cancer. So this particular field is called oncology, and we are in the field of oncology, and that's why we will be learning some aspects with respect to oncology. Now, I'm sure all of you will know when the cell has to divide, it does in two ways. One is, of course, mitosis and meiosis. But let us just understand why the cell division is required. See, when a new baby is born, of course, cell division is required because the child has to grow, the organs have to grow, the size of the child has to grow, hands, limbs, legs, all have to grow. So you require cell division, right? So which is a very natural process. When a child is growing, cell division has to take place. But when an adult is already formed or is already, when a person is an adult, then why you require cell division? Okay. So in our day-to-day lives, also we require cell division for the normal functioning of the body in a proper and a structured way. See, because every day there is wear and tear in our human body. Some cells uh, grow, some cells divide, and some cells die also. So, to replenish the cells which have died or which have got damaged, you require cell division. Okay, so this is a natural process which is happening in our day-to-day life. Cell division is happening continuously in every human body, and there are certain conditions, certain exigencies which come up, like if you get hurt, if your body starts bleeding, then also you require cells to multiply and take care, because the bleeding which has taken place has to stop. So there has to be coagulation. So there are multiple cells which are involved in cell coagulation also. So there also you require cell division and cell multiplication, right? So this is a normal phenomena which happens in every human body. But when this goes haywire, if it is not in a controlled manner, okay, and you have abnormal cells, then that will lead to cancer. Okay, so that is how cancer develops, when there is abnormal cells and they multiply in huge number. So then they can turn into cancer, and not necessarily all the abnormal cells which have which are dividing and multiplying will lead to cancer, but it can lead to abnormal mass, which can be tumor. And when it becomes malignant, that means when it starts spreading, then it becomes cancerous. Okay, so this is also something which is happening every day in every human body. So there are normal cell divisions, and there are some cancer cells which are formed every day in every human body. But if these abnormal cells or this cancerous cell growth which has taken place in our human body is not taken care of by the person's human body, then it leads to cancer. Okay, so there is a normal mechanism in every human body to take care of all these cancer cells also. But if the body fails, then the patient can tend to develop cancer, and then if it becomes malignant, if it starts spreading, then it can have very, very adverse effects. So this is a normal phenomena. But what we need to understand, this cell division which is taking place occurs in two ways. One is mitosis and one is meiosis. Kimia, is my speed okay? Kimia, is my speed okay? Okay, okay.
Yes, your speed is okay. Yeah.
Okay, thank you. So if you all have any issues, you all can let me know. I can interrupt in between. Yeah, I mean, you all can interrupt me in between. Yeah. Okay. So basically, when you talk about cell division, it takes place in two ways. One is mitosis and one is meiosis. I'm sure all of you all, because I have seen everyone's profile, all of you all are B farms. So all of you all understand what is mitosis and meiosis. Just to explain you or highlight few of the things, mitosis basically takes place in all the somatic cells. So what do you mean by somatic cells? The cells, all the cells in the body except the reproductive cells are called as somatic cells. So you have reproductive cells and you have somatic cells in the body. So all the somatic cells in human body will multiply with the help of mitosis. Now what happens in mitosis? From one parent cell, two daughter cells are formed, and the two daughter cells which are formed are exactly the same replica of the parent cell. So what do you mean by exact replica? They are identical to the parent cell. So the entire cell organelle which was there in the parent cell, even the two daughter cells have the same cell organelles. And the number of chromosomes which are present in the parent, that means 23 pair of chromosomes in the parent cell, even the daughter cells will have 23 pairs of chromosomes. So they are exactly identical and true replicas of the parent cell. So basically, parent cell is also diploid. Diploid means it has got uh, two pairs of chromosomes, so they are called as diploid, and n is equal to 23. That means uh, 23 pairs of chromosomes, and even the daughter cells are diploid, right? So in mitosis, what happens? From one parent cell, two daughter cells are formed. These two daughter cells are exactly identical to the parent cell, and they are also diploid in nature. So we have to understand mitosis because we are going to understand mitosis because this is going to actually help in understanding of all the products, right? We will not be going in too much of detail with respect to meiosis. Meiosis basically is for the reproductive cells. Just one second. Meiosis is basically cell multiplication which is seen in the reproductive cells. Basically, what happens in meiosis is you have reproductive cells in the testes and ovaries. Meiosis actually occurs in the testes and the ovaries, and these are the organs which are actually responsible for reproduction. So when the sperm cells and the egg cells have to multiply, they undergo a process called meiosis. And meiosis, what happens? The parent cell divides into two daughter cells, but they have just half the number of chromosomes of the parent cell. So this is what is happening in meiosis. We don't have to go into much of detail because when we talk about chemotherapeutic drugs, we have to understand mitosis, and we will not be going into meiosis because meiosis itself is a very big chapter, and in oncology we don't deal with meiosis, right? So we will understand what exactly is happening in mitosis. Just one second. Okay, so now on my screen you will see cell cycle. Mitosis is a procedure in which the somatic cells are dividing. That means all these cells, other than the reproductive cells in the human body, divide with the procedure called mitosis. But mitosis also occurs in a phase manner, okay, and the different phases come under the cell cycle. So we have to understand the cell cycle and what actually is happening in each phase of the cell cycle. We need to understand that. So basically, if you see here, you can see there are five different phases of a cell cycle. One is the G1 phase, the other is the S phase, then you have the G2 phase, you have the M phase, and then after that you can see the G0 phase. Okay, so these are the five different phases of a cell cycle. We will understand each one of them in detail. So basically, when a cell has to divide from one parent cell to two daughter cells, there are different steps involved, and that is being covered in this cell cycle. As we say, cell mitosis or cell division is taking place. A parent cell is getting divided into two daughter cells. There are so many steps involved. There are so many enzymes, so many proteins, so many chemicals involved. Only after each one of them starts working in their right manner, you have cell mitosis taking place. Okay, so that's why understanding each phase or each step is very important for us. The first phase, as you can see, is a G1 phase, also called as a cell growth phase. So here what happens? Basically, the cell is preparing itself for cell multiplication. And when you talk about mitosis, what happens first is the DNA has to duplicate itself. So first DNA duplicates itself and then the remaining cell organelles divide and then two daughter cells are formed. So in the G1 phase, what happens is all the organelles which are required are getting formed, and even the cell organelles which are present will be growing, and the cell is preparing itself for the S phase, that is the DNA replication phase. But when DNA replication has to take place, the cell has, I cannot see your video. Hello, is there a network issue, Kenya?
Don't worry, I'm here.
Okay, okay, okay. Because if I see you, then I can make out that everyone is listening. Yeah. Because I can see
No
only you,
problem.
I can't see the others. Yeah. Okay. So
By the
when
way, I can like make like this. Yeah.
no, anyhow, whatever you feel is okay. I don't mind. Okay. Yeah. Now I can see all of them.
Okay.
Okay, okay. So when DNA synthesis has to take place, the cell has to prepare itself in many ways because more energy is required for DNA replication. So cell has to synthesize that energy which is required. Various types of proteins and enzymes are required for DNA replication. So in the G1 phase, the cell is preparing itself by making more energy, by making the required proteins, by making the required enzymes for DNA replication. So in the G1 phase, the cell is growing, plus it is preparing itself for the DNA replication, that is the S phase. Here you can see I have mentioned there is a repair of thymidine dimers. See, DNA is made up of purines and pyrimidines. They have various bases. You have thymidine base also in the DNA. But here, there is repair of the thymidine dimers. Because what happens in every human body when it is exposed to some UV rays, there are sometimes thymidine dimers get damaged. Okay, so that correction also takes place in the G1 phase. So with the help of certain enzymes like nuclease enzyme, the thymidine dimers which have got damaged also get corrected in the G1 phase. So in the G1 phase, basically cell grows. There is synthesis of different types of proteins and enzymes, and also body synthesizes more energy which will be required for the DNA replication to take place. Plus the thymidine dimers which generally get damaged after exposure to UV rays, they also are repaired in the G1 phase, right? So after the G1 phase, you have G1S checkpoint, as you can see here. There's G1S checkpoint. Now, what is this G1S checkpoint? When the cell is preparing itself in the G1 phase, the cell has to enter the DNA replication phase. The DNA replication phase is a very, very important phase because there are so many steps involved in DNA replication. Plus, DNA replication is the most important phase in cell cycle. Okay, so before the cell enters actual DNA replication in the G1S checkpoint, the cell will check whether the right proteins have been formed, whether the right enzymes have been formed, the required amount of energy which will be used is already present or not, whether the thymidine dimers have got corrected or not. Okay, so all this will be checked after the G1 phase and before it enters into the S phase. Okay, so this is a checkpoint to see that the cell is fully ready for the S phase. So you have the G1S checkpoint where the cell is checking whether it is prepared fully for the S phase. Then after the cell passes the G1S checkpoint, it enters into the S phase. Now what's happening in the S phase? In the S phase, you have the actual DNA replication which takes place. Here you have the actual DNA replication which takes place. So basically here the DNA will duplicate itself. So the number of DNA gets doubled. Okay. And after the DNA duplicates itself, the number of chromosomes of course remain the same. So in this step where the DNA duplication is taking place is called as the S phase or the synthetic stage. And here you have the synthesis of histone proteins also. I'm sure all of you know that DNA is helically shaped, right? And each chromosome is containing one DNA, and DNA is also quite long, so it has to adjust and come within the chromosomes. So how this happens? With the help of these histone proteins. Histone proteins will give us support so that these DNA take a proper shape. So basically, these histone proteins behave like a mold. Okay, and in this mold, the DNA will go and take its proper shape and then it can fit inside the chromosomes. So you have the histone proteins also which are getting formed in the synthetic phase. So in the S phase, the DNA gets duplicated. That means the genetic matter is completely duplicated in the S phase. So now the cell is ready to enter into the phase. Now you can see the G2 phase here. After the DNA duplication, the cell is entering into the G2 phase. What is happening into the G2 phase? The cell is preparing itself for cell mitosis. That means in the G2 phase, the cell will prepare itself so that the actual cell mitosis or the cell can actually divide into two in the mitosis phase. So it will prepare itself in the G2 phase. Like how in the G1 phase, cell was preparing itself for the S phase. Here in the G2 phase also, the cell is preparing itself for cell mitosis. Again, there are many proteins, enzymes which will be required for the cell mitosis, which are all being formed in the G2 phase. So again, there is cell growth. All the proteins and enzymes which are required are also getting synthesized here, which will be required for the cell mitosis. Plus, what happens in the S phase, the DNA had duplicated. So you have double number of DNAs. But when you have uh, duplication of DNA is already there, but the other cell organelles also have to divide and form into two cells. Okay, so how will that happen? First, the genetic matter or the DNA which has duplicated has to get divided because one set will go to one cell and the other set will go to the other cell. So how that is done? That is done with the help of the spindle formation. You have spindles, the mitotic spindles get formed. Now these mitotic spindles are thread-like structures which polymerize and they form long strands. Now with the help of the spindles, the DNA which had duplicated gets attached and one set moves to one side and the other set moves to the other side. Okay, so you have spindle formation in the G2 phase and all the cell organelles. See, every cell has got multiple cell organelles. It has got ribosomes, it has got mitochondria, it has got Golgi apparatus. So all that also has to duplicate. Only when they duplicate, the cell multiplication can take place. So in the G2 phase, the cell is preparing itself for cell mitosis. Plus you have the spindle formation, all both, both the groups of DNA are pulled to two different sides, and all the cell organelles are getting duplicated in the G2 phase. Okay, so in the G2 phase, the cell has prepared itself for the M phase, that is the mitotic phase. Now you have another checkpoint, that is the G2 M checkpoint. Here again, the cell has to see whether the cell organelles have got duplicated or the spindle formation is proper. So the cell again checks whether the cell is fully ready to enter into the M phase. Okay, so this is the G2M checkpoint where the cell is checking whether the cell is actually ready to enter into the M phase, that is the mitosis phase. Now, what had happened in the S phase? The DNA had duplicated. In the G2 phase, mitotic spindles have formed and they have taken the genetic matter of the DNAs to two different sites. Okay, and in the S phase, the original cell's nuclear membrane had dissolved. Only then the DNA could move out. But what will happen in the M phase? Again, the nuclear membranes will be formed because two nuclear membranes will be required for two different cells. What is nuclear membrane? Nuclear membrane is the outer lining of the nucleus, and the entire genetic matter of the DNAs and the chromosomes are inside the nucleus. So when two new cells have to be formed, again the nuclear membrane starts coming. Okay, so now this nuclear membrane will start coming in the M phase. The mitotic spindles which had formed and which had attracted the DNA, where exactly the mitotic spindles originate or they start forming? They start forming at one point called centrosome. So from centrosomes you have the mitotic spindles which are coming out, and these mitotic spindles go and attach to the DNA strand or to the chromosomes. The point where they attach to the DNA or the chromosomes is called as kinetochores. So these from centrosome, you have the mitotic spindles which are coming out, and these mitotic spindles attach the DNA at a point called kinetochores. Okay. Now in the M phase, what happens? The mitotic spindles are attached to the kinetochores, that means to the DNA. Now they have to take back the DNA because one cell will get 23 pairs of chromosomes and the other set will get 23 pairs of chromosomes. So how the DNA gets attracted? The mitotic spindles which had formed in the S phase, they have to be pulled down. So there is compression, and the tension which was there originally, that has to be released. So then these mitotic spindles get depolymerized and they become small, and when the tension is released, these mitotic spindles pull back all the DNA strand to two different sides of the cell. So the DNA has 23 pairs of chromosomes have come to one side. Yeah. Anything, Kimia? I can
Now you
proceed.
can continue.
Okay. And the other 23 pairs of chromosomes have moved to the other side. Okay. And again, now what happens? The nuclear membranes start coming. And then what happens here when the nuclear membranes start coming? The entire genetic matter is covered inside the nuclear membrane. So a nucleus is formed, and all the cell organelles which were present in the cell which had duplicated in the G2 phase will also go to two different sides, and then after the entire genetic matter and the cell organelles have gone to two different sides, you have cytokinesis where the cytoplasm also divides, and then you have the formation of two new cells which are exactly identical to the parent cell. So this is happening in the cell mitosis phase. Okay. Cell must be a certain size before daughter cells would be large enough to function normally, must have enough nutrients, so that is checked in the G2M phase, and you have a M checkpoint where again they see whether the cell can actually undergo a split, and after the checkpoint is cleared, then cell can divide, all the cell organelles divide, and after the cell organelles divide, even the cytoplasm divides. So after the formation of the nuclear membrane, the cell membrane also forms and divides. So you have two new cells which are formed, which are identical to the parent cell, and each cell will have 23 pairs of chromosomes, will have two nucleus, will have all the cell organelles like Golgi apparatus, your mitochondria, cytoplasm, nuclear membrane, cell membrane. Okay, all the cell organelles will be there in the two new daughter cells which have formed. Now here you can see after the cell mitosis or the cell division which has taken place, again the cell can enter into the G1 phase or it can go into the G0 phase. Now what is the G0 phase? In G0 phase, basically, the cell can go and rest. So the resting phase is called as G0. That means after the mitosis has taken place, two new cells have formed, the cell can easily go to the G0 phase and rest. So there will be no cell multiplication, no cell division in the G0 phase. Okay, but the cells have got a choice. They can enter the G0 phase or they can again enter the G1 phase. That means again they can undergo the cell mitosis phase. That means again they can go to the G1 phase where they will prepare its cell for cell mitosis. So the cells can either go to the G0 phase, that is the resting phase, or they can go to the G1 phase where they can again enter the cell cycle and start further cell multiplication. Now you will have a question why you have G0 phase. Now this G0 phase, though it is called a resting phase, what happens here is some of the cells which have multiplied and formed into daughter cells, they have to start functioning in a specific manner. Say today we have got so many different cells in our body. Uh, when you talk about blood cells, you've got RBCs, you've got WBCs, you've got platelets, right? But originally they were a stem cell, or you can call it as a pluripotent cell. Okay, from this pluripotent cell, they transform in such a way that some form RBCs, some form WBCs, some form platelets, right? But how this happens? This happens with the procedure called cell differentiation. Now, what is cell differentiation? Here, the new cells which have formed, they will take up a new structure, they will take up a new function, and they will start functioning in a specific manner. So, from the parent cell, which was like a stem cell or which was also called as a pluripotent cell, they will take up new structure so that they can perform a specific function of either a RBC or a WBC or a platelet, or some may form neurons, some may form lung cells, some can form brain cells, all different. So this is how you have different cells getting formed in our human body, which can perform specific function. So this G0 phase is also very important. Here the cell is not undergoing any multiplication, but it is undergoing a process called cell differentiation, right? And when it goes to the G1 phase, then of course it will again start the cell multiplication. So understanding the cell cycle is very important because our chemotherapeutic drugs will act on different phase of the cell cycle. Some might act on the G1 phase, some will act on the S phase, some will act on the G2 phase, some will act on the M phase, or some can also act on in between the checkpoints like G1S phase or G2M phase. Okay, so that's why we have to understand what is cell cycle and which are the different phases of the cell cycle. Okay. Now, as I told you, it is important for us to understand the cell cycle. Why? Because the chemotherapeutic drugs, that means the drugs which are used for treating cancer, are cell phase specific. What do you mean by cell phase specific? that they act on some particular phase of the cell. Like some may act on the G1 phase, some may act on the M phase. So they are called as cell cycle specific. But there are some chemotherapeutic drugs which are cell cycle non-specific. So what do you mean by that? Cell cycle non-specific chemotherapy drugs are most effective against actively dividing cells, but also effective against cells in G0 stage. So cell cycle non-specific are chemotherapeutic drugs which can act on any phase of the cell. Okay, and they can act on all phases of the cell also, including the G0 phase. So cell cycle specific are specific to a particular phase, but cell cycle non-specific drugs are the drugs which can act on any phase of the cell cycle, including the G0 phase. Okay. So understanding this is very important. Some drugs are cell cycle specific. They act on one particular phase. Some drugs are cell cycle non-specific. They can act on all the phases of the cell cycle, including the G0 phase, right? Now, we just understood what is a cell phase or the cell cycle. We also saw what is cell cycle specific, what is cell cycle non-specific in chemotherapeutic drugs. But as I told you, when a patient gets cancer, there are various types of treatment options available for a patient's treatment. Okay, depending upon the patient's condition, the stage, the size, the treatment models are decided. But just to give you some idea, for treating solid tumors, you have cancer treatment options like surgery, where you actually do the surgery, remove the cancerous growth, and remove all the adjacent tissues and nodes which are affected, and remove the cancerous growth. So that is one option. But surgery is possible only when you have solid tumors. Then you have one more option like chemotherapy, where we will be focusing on, where you give chemotherapeutic drugs to kill all the cancer cells. Okay, chemotherapy can be given uh, for treating cancer or can be the first line of treatment also. But chemotherapy also can be given after surgery. Why after surgery? Because when a patient undergoes surgery, even you try to remove the cancerous cells which were around the cancerous growth, but still some cells remain in the human body. So to kill or to manage the cancer cells which remain after surgery, you give chemotherapy, because chemotherapy generally is systemic in nature, so it will take care of all the cancerous cells which remain even after surgery. Okay, so that's why it can be given after surgery. It can be given for treatment also. Okay, and chemotherapy can be given along with radiations also, plus it can also be given after radiation or even before radiations also. So the combination is decided by the oncologist. Then you have radiation therapy. What happens in radiation therapy? Patients get high powered energy beams like X-rays or proton rays, which are given to the patient at the site where the cancer is there. Okay. So in radiation therapy also, you have various types of radiation therapy like external beam, brachytherapy, and stereotactics. We will not go into too much of details because we'll be restricting our discussion to chemotherapy. But radiation therapy has also progressed. There are multiple options in radiation therapy also. Even the radiation therapy can be regulated. You can give high dose, you can give low dose, you can give fractionated dose. That means you give small doses at certain frequencies. You give small dose but increase the frequency, or at times in cyberknife technology and such things, you give high beam, pinpoint, but just one shot, you give one shot of radiation. Okay, so you have those types of radiation therapy also. So some of the radiation therapy have got some side effects also associated with it. And sometimes radiation therapy is not possible because if the cancer is deep seated, the radiation therapy is not advisable. and even doing radiation therapy for blood cancers or lymphomas sometimes becomes difficult. So then chemotherapy is opted, or sometimes combination is given, chemotherapy with radiation therapy, or first you give surgery and then do radiation therapy. So different types of combinations can be adopted. Okay. Now, chemotherapy, as I told you, is something which we will be studying in detail. Basically, if you read various textbooks and certain protocol booklets, or if you read the way the doctors write, there is an abbreviation which is used for chemotherapy. It is called as CTX. Sometimes CTX is all in caps. Sometimes it is like CT and X is small. So this is the abbreviation used for chemotherapy. Now, what is chemotherapy? It is a cancer treatment that uses one or more anti-cancer drugs, which are also called as chemotherapeutic agents, as part of a standardized chemotherapy regimen. I'm sure by now all of you would be knowing when we talk about chemotherapy, we talk about anti-cancer drugs, which are also called as chemotherapeutic drugs. So sometimes a patient gets a single drug, or sometimes patient gets a combination of drugs. Okay, so in chemotherapy, sometimes patient will get a single drug, and very often the patients will get multiple drugs. So what happens when a patient is getting multiple drugs? It is called as a protocol or a regimen. When you have two or more than two drugs being given as chemotherapy for treating a cancer patient, then that is called as a protocol or a regimen. Okay, so you might have seen for breast cancer, you have got so many protocols like AC regimen or AC protocol, CMF regimen or CMF protocol. Okay, similarly for multiple myeloma, there are different types of protocols. So there are various protocols being used for treating different types of cancer. Now, when you talk about chemotherapy, chemotherapy sometimes can be given as a curative. What do you mean by curative? It can be given to treat the patient from the disease or from cancer. But very often, and I would say most often, chemotherapy is given to prolong the patient's life. That means to increase the patient's lifespan or to reduce the symptoms of the patient. Now, when you talk about reducing the symptoms of the patient, that becomes a palliative chemotherapy. Okay, so chemotherapy can be given to treat the patient or so that the patient gets free of cancer. Sometimes chemotherapy is just given to prolong the life when treatment is not possible, then the patient's life is prolonged. So give a chemotherapy, increase the lifespan of the patient, and at times it is given to reduce the symptoms, because we know patients who are having cancer can suffer from pain, may have diarrhea, may have nausea and vomiting. Okay, so you give chemotherapy so that the patient's quality of life improves, and that is called as palliative chemotherapy. So basically, what is actually done in chemotherapy, when you see the different types of chemotherapeutic agents, you will see most often chemotherapy is inhibiting mitosis, which is also, just one second, one second. Yeah. Sometime back, we just, I'm sorry, there was a phone call. Okay, so I'm just starting again. Yeah. We had just seen in my initial slide what is cell mitosis and the different phases in cell mitosis, right? Why we had studied that? Because chemotherapy or chemotherapeutic drugs act on the different phases of the cell. So basically, chemotherapeutic drugs inhibit cell mitosis, which is also called as cell division. Okay, because they are affecting one or the other phase of the cell, or maybe all phases of the cell. Therefore, we say chemotherapy actually inhibits. Therefore, we say that chemotherapy actually inhibits cell division or cell mitosis, and thereby it, of course, damages the DNA also. And one most important thing is the chemotherapy can be systemic or it can be loco-regional. When we talk about systemic, which is majority of the chemotherapies are systemic in nature. What do you mean by systemic? That they go and affect each and every cell in the human body because they are introduced in the human body through the bloodstream. Okay, and they can be given intravenously if it is a liquid chemotherapy. If they're given orally, then the patient takes it through the GI tract, but it goes through the bloodstream and reaches each and every cell in our human body. Therefore, it is called as systemic in nature. Okay, and at times when it is given at the site where the tumor is there, it is called as locoregional. Okay, but majority of the chemotherapies are classified under systemic chemotherapy only. Uh, of course, this is altogether a different subject, but I'll explain you the advantages and the disadvantages of systemic therapy. Basically, when the patient is getting systemic therapy, this chemotherapeutic agent is getting introduced in the bloodstream. Therefore, it reaches each and every cell of the human body. Therefore, if you see what will happen, there are side effects of chemotherapeutic drugs, as we know. So, when a patient gets systemic therapy, the side effects also will be systemic. It will be seen all across the body, right? But if you have chemotherapy which is loco-regional and is given only at the site where the tumor is there, then the side effects will also be, at that site itself. Okay, so the damage or the side effect will be less, whereas in systemic chemotherapy, the side effects will be more because it is reaching everywhere. So it can, of course, damage the cancer cells and it can damage also the normal cells. Okay. Now, what is the principle behind chemotherapy? I'll just explain you. Basically, chemotherapeutic drugs are designed in such a way that they can damage or they can kill the fast multiplying cells. Okay, and as we know, cancer has got abnormal cells, plus these cancer cells are fast multiplying. That means they multiply very fast in comparison to the normal cells. Normal cells do not multiply that fast, but abnormal cells or the cancer cells multiply very fast. Therefore, this chemotherapeutic drugs action is to inhibit or to kill the cells which are fast multiplying. Okay, so that is the principle behind chemotherapeutic drugs, that they will damage or they will inhibit the fast multiplying cells. Therefore, they are damaging or killing the cancer cells. But that does not mean that they will not damage or they will not cause any effect to the normal cells which are not multiplying very fast. They will damage, but that will be to a lesser extent. Okay, so they will kill all the fast multiplying cells, that means the cancer cells, but they will also damage the normal cells which are present in a human body. But in our body, other than the cancer cells, there are many cells which are very, very fast multiplying, like your blood cells, because in the bone marrow you require RBCs, WBCs, platelets very often, so they multiply very fast. They are required daily. They multiply very fast. So these chemotherapeutic drugs can affect the bone marrow where the cell multiplication is very fast. Okay. Plus in the GI tract you have the mucus lining. Now, this mucus lining is required because you take food which has to slide down. So and plus you have the saliva in the mouth which actually helps in the digestion. So here there is continuous production of saliva. So these are all fast multiplying cells. But when chemotherapy is given, they will also damage these cells because they are fast multiplying. So they can damage the salivary glands which are present in the mouth. They can or damage the mucosal lining. That's why you have GI irritation. Patients suffer from nausea and vomiting. Plus, if you have seen, the cancer patients generally lose hair. That is called as alopecia, because hair cells also multiply very fast. Therefore, these chemotherapeutic drugs can damage the fast multiplying hair cells. Therefore, you have heavy hair fall when a patient is taking chemotherapy. Okay, so this is how chemotherapeutic drugs act. They act on the fast multiplying cancer cell, plus they can damage the fast multiplying other cells which are present in the human body. So that is the principle behind chemotherapeutic drugs.
And you have to unmute yourself. You have muted. Yeah.
Okay. Yeah. So basically, what is the aim of chemotherapy? They can, as I have already told you, they can be given to cure the patient. They can be given to prolong the patient's lifespan, and they are also given as palliative care, where they will manage the side effects and improve the quality of life of the patients. So this is the purpose of giving chemotherapy. Now, what exactly are chemotherapeutic drugs? I have actually explained you, but I'll just take you through these slides once again. Chemotherapeutic drugs are cytotoxic in nature because they are killing the cells, plus they can cause toxicity to the normal cells. Also, they kill cells usually by disrupting the essential normal functions of proliferating cells. The most important function is DNA synthesis, so they can damage the DNA synthesis, and then they can kill the cancer cells. Because cancer cells proliferate more rapidly, as I told you, than the normal cells, cancer cells are more susceptible to chemotherapy. Why? Because chemotherapeutic drugs will act on the fast multiplying cells, and cancer cells are fast multiplying cells. Therefore, the cancer cells get damaged when chemotherapeutic drugs are being given. But as I told you, they can also damage the normal cells, which also multiply very fast. That's why you see so many side effects associated with the chemotherapeutic drugs. They increase the concentration of the drug near the tumor if it is locoregional in nature. Now, basically, Shiva, can you hear some disturbance?
Yeah, there is outside disturbance. Drum is there.
Yeah, there is a lot of
But I think
background.
it is not.
It's not
We need to
disturbing,
ask
right?
them. We
Okay.
need to ask them. Yeah.
No, you are not getting disturbed, no? So I
No,
same
I am
thing.
not getting disturbed.
Fine,
No.
fine. So then you'll proceed. Now, I told you that when you get chemotherapeutic drugs, they can also affect the fast multiplying normal cells. So now let us understand the toxic effects of chemotherapeutic agents. Chemotherapeutic drugs are cytotoxic. They can differentiate between tumor cells and normal cells because tumor cells are fast multiplying. Normal cells generally don't multiply very fast with some exceptions. Hence, normal cells are also damaged by chemotherapy, but at a slower rate. In oncology, the side effects which you see, which are associated with the chemotherapeutic drugs, are called as toxic effects, or it is also called as toxicity. Okay, so here in oncology, you will see the usage of adverse effects or toxic effects being used when we talk about all chemotherapeutic agents. Toxic effects caused by chemotherapy tend to be systemic. Why systemic? If the chemotherapy that you're given is systemic in nature, then the side effects also will be systemic. That means they will be seen all throughout the body. If toxic effects are intolerable, chemotherapy doses must be adjusted or chemotherapy must be discontinued. The one thing good about chemotherapy is, though the side effect will be seen all across the body if the chemotherapeutic drug is systemic in nature, right? But if the patient who's taking chemotherapy finds that the side effects are more, if the patient is suffering from severe diarrhea or if the patient is suffering from severe nausea and vomiting, then the dose has to be adjusted. So what do you mean by dose adjustment? The dose which was recommended has to be reduced. Okay, so sometimes the dose is reduced so that patient does not suffer from the toxic effect. Sometimes the dose has to be discontinued. That means the therapy which you were given, that has to be stopped. Okay, or sometimes you increase the gap. You have given a chemotherapeutic agent or multiple agents, you see some toxic effect, then you don't give the chemotherapy. You give a gap between two chemotherapies so that the patient can come back to normal and the patient's blood counts and the other parameters can come back to normal, and then you start giving chemotherapy. So there are various ways in which the chemotherapy can be modulated and regulated. So sometimes you decrease the dose, sometimes you discontinue the dose. If the patient is not able to tolerate, you discontinue that chemotherapy and you can switch to some other chemotherapeutic agent. Sometimes you increase the time span between two chemotherapies so that the patient's body comes back to normal and the patient starts tolerating the chemotherapy. So there are various ways in which chemotherapeutic agents dose can be regulated. Yeah. So we had seen what is cell cycle specific and what is cell cycle non-specific. Just to take you through it again, cell cycle specific means that the chemotherapeutic drug is acting on one of the phases of the cell cycle. If it is cell cycle non-specific, that means it is acting on all the phases of the cell, which includes even the G0 phase. Like it can act on S phase or the G2 phase, or it can act on M phase. Okay. Now, when you come to chemotherapeutic drugs, which are cell cycle specific, till now what we have seen is basically how the cell multiplies by the way of cell mitosis. We saw cell mitosis in detail. Then we also saw the different phases in cell mitosis. Uh, then what is the significance of understanding cell mitosis? We saw that. We also saw what are chemotherapeutic agents and what is their role, how the dose can be regulated, how they act. Now let us understand what are the different types of chemotherapeutic agents. We have seen chemotherapeutic agents. Now let us understand the different types of chemotherapeutic agents. We saw the different types of treatment which are available for treating cancer patients, like surgery, you have radiations, and it can be a combination also. But we will not go into the depth of surgery and radiation therapy now because we are dealing with chemotherapeutic agents, right? So there are different types of chemotherapeutic agents. The first one which we will be seeing is an antimetabolite. Okay, from the name itself, you can make out that this is anti-metabolite. So it is anti to a metabolite. Now, first and foremost, this chemotherapeutic agent acts on the S phase. What is the S phase? It is called as the synthetic stage or the stage where the DNA replication takes place. So your antimetabolites are going to act on the S phase. Now, what are antimetabolites? A chemical that inhibits the use of a metabolite, which is another chemical that is part of a normal metabolism. Okay, basically, many metabolites are required for the normal functioning of the cell cycle, which can be either in the form of enzymes or proteins or folic acid. So all these metabolites are required for the normal cell cycle to function. Now, these antimetabolites, when they enter, or when these chemotherapeutic agents they enter, they resemble these antimetabolites. Okay, so they will resemble the vitamins, or they will resemble the purines, or they will resemble the pyrimidine bases. So if they resemble, what happens? They have already entered into this human body or in the cell. The cell will feel it is a normal purine or it is a normal pyrimidine, so it will take it up. Okay. So instead of the normal purine or a normal pyrimidine, which is required for the cell to multiply, you have an antimetabolite which is come, which is structurally similar. Okay. So something it has come which is not the actual purine or a pyrimidine, but it has already entered. So what will happen? The normal cell function will not take place because you don't have the right vitamin or the right purine or a right pyrimidine base which has entered. Okay, so the normal cell functioning will be disturbed. So these antimetabolites, what they do they are acting on the S phase, though they will represent vitamins, purines, and pyrimidine, they will enter in the S phase, they will be taken up by the cell, but they will not allow the DNA replication to take place. So these antimetabolites will act as substrates for DNA synthesis enzymes and compete with natural substrates that are used in DNA synthesis, such as purines and pyrimidines. So this process where you don't have the right purines, pyrimidines, or folic acid or vitamins, then what happens is the abnormal antimetabolites are present, the normal ones are not allowed to be taken up. This process is also called as masking. Okay, so what do you mean by masking? The right ones are not allowed to come, but the wrong ones are present. So this procedure is called as masking. So because of masking, the normal metabolites are not given to the cell and the normal functioning of DNA replication does not take place. So that is a simple mode of action of antimetabolite, and this is happening in the DNA replication phase. If this is happening in the DNA replication phase, what happens? The DNA cannot replicate. If the DNA cannot replicate, what will happen? The duplication of the DNA will not take place. If the duplication of DNA does not take place, then obviously cell cannot multiply because the DNA itself has not duplicated. The cell multiplication will stop. So the original cell will also die, plus the further cell growth will also be taken care of. Okay, so this is how an antimetabolite actually acts. There are different types of antimetabolites. We will not uh, learn about all types because we are concerned with only few of them. There are different types like citrabine, you have mercaptopurine, you have 6-thioguanine, you have 5-fluorouracil, you have methotrexate. Because some of them are purine analogs, that means their structure is similar to purines like 6-thioguanine, 5-fluorouracil, which resembles thymidine synthetase. Okay, and it is actually responsible for the formation of thymine, which is actually required for the DNA synthesis. But we will not go into the details because we don't have products in those categories. We will be considering methotrexate because we have methotrexate with the name Mthex with us. So what is actually methotrexate? It is a folic acid analog because its structure is similar to folic acid, therefore it is called as folic acid analog. What it does, it will inhibit the enzyme dihydrofluorate reductase, which inhibits the folic acid synthesis, which is essential for formation of purines and thymidine dilate. Thus, it inhibits the synthesis of RNA and DNA. So, what you have to understand here is basically methotrexate is a folic acid analog. Folic acid is required for the synthesis of RNA and DNA, which is an essential component for DNA replication to take place. Okay, so folic acid is required, folic acid synthesis is required for the DNA replication, which is essential. Now this folic acid analog has entered, methotrexate has entered, which is a folic acid analog. So it will go and mask. So because of which the actual folic acid will not be present, which is actually required for the RNA and DNA synthesis to take place. So if you don't get the right folic acid for RNA and DNA synthesis to take place, the RNA and the DNA synthesis cannot take place because the right folic acid is not present or is not freely present. Therefore, the RNA and DNA synthesis will not take place. If RNA and DNA synthesis is not taking place, obviously cell will not, obviously DNA replication will not be there. If DNA replication is not there, then further cell cannot multiply itself. So that is a simple mode of action of folic acid analog, and one of the drug which is a folic acid analog is methotrexate, which we will be seeing when we go to MThex. Then you have various other antimetabolites like pamitrexate, which actually inhibits three enzymes which are used in synthesis. But since you all don't have pamitrexate at the moment, we will not go into the depth of pamitrexate, capecitabine, gemcitabine, and hydroxyurea. Yeah. Now, hydroxyurea basically inhibits enzyme called ribonucleotide reductase enzyme, which inhibits DNA synthesis. As I told you, there are so many enzymes which are required for DNA synthesis. One of the enzymes which is required is ribonucleotide reductase enzyme for DNA synthesis to take place. Now, when hydroxyurea is given, it inhibits the enzyme ribonucleotide reductase. It will inhibit this enzyme. Therefore, if the ribonucleotide reductase enzyme is inhibited, it will not allow DNA synthesis to take place smoothly. Okay. Therefore, the role of hydroxyurea is to inhibit the enzyme ribonucleotide reductase. Okay. If this enzyme is inhibited, then obviously cell cannot undergo DNA synthesis. If DNA synthesis will not take place, then obviously cell cannot multiply. The original cell also dies and further cell multiplication is also stopped. Okay. So we will be studying about hydroxyurea, we'll be studying about methotrexate in detail when we go to the product.
And you have muted again.
When I change the slide, it gets muted. Okay. Yeah. Now, when you, in the previous chemotherapeutic drug, which was antimetabolites, we saw that they are acting on the S phase. Okay. Now we will see another chemotherapeutic agent, which is cell cycle specific, and it is acting on the G2 phase. So G2 phase is the phase where the cell is actually preparing itself for cell mitosis.
Again, you have muted, man. Unmute yourself. Unmute yourself.
Oh God, how is this happening? 1 second. Yeah. Yeah. Now to understand this particular chemotherapeutic agent, we have to understand a very simple thing. Though this diagram appears to be very, very complicated, I will just simplify it for you. You don't have to understand each and every part of this diagram. Basically, DNA, if you see, is helically shaped. This portion you will see, DNA has got two strands. They are helically coiled. Okay, so they are coiled like this. But when DNA replication has to take place, these two DNA strands have to unwind themselves. What do you mean by unwinding? That means there has to be uncoiling. So when they uncoil, two separate strands of DNA will be formed. Right? So when they uncoil, two separate strands are formed, but they have to duplicate. So one more strand will come and join one of the strands, and there'll be another strand which will be formed, which will come and join the other strand. So then you will have two DNA which will be formed. but you have two strands on each side, but again they have to coil and form into DNA strand. So this is a simple way in which the DNA synthesis take place. There are various enzymes, various proteins which are involved. But what we have to understand, when the DNA strand has to uncoil itself and coil itself, there are enzymes called as topoisomerase. Okay, so topoisomerase enzymes help in the coiling of in the uncoiling of the DNA strand, and they also help in uncoiling of the DNA strands. So this topoisomerase enzymes manages the coiling and uncoiling of the DNA strand. How it does? It basically causes a cut in the DNA strand, a single cut or a double cut, okay, because of which the tension is released, and then the DNA can uncoil and coil itself. And after it gives a cut, the DNA strand uncoils itself, but again it has to coil back, so it helps in joining also of the DNA strands. So this is how the topoisomerase enzyme actually acts, and it acts on the G2 phase. Not muted. Yeah. Now here, if you see, I told you topoisomerase enzyme is the one which helps in the coiling and uncoiling of the DNA strand, either by causing one cut in the DNA strand or by causing two cuts in the DNA strand. There are two types of enzyme. There are two types of topoisomerase enzyme. One is topoisomerase 1 and one is topoisomerase 2. Okay, so topoisomerase 1 enzyme, what it does, this is a type of enzyme which cuts one strand of a DNA, which is double helix, and relaxes so that they can uncoil, and then it will again join. It will re-anneal, that means it will help in the reformation of the double strand DNA molecule. Okay, so first it will give one cut to the DNA strand, so it will uncoil itself. Later on, it will join the DNA strand so that the double helix can be formed. So this is the role of topoisomerase 1 enzyme. Now, what does topoisomerase 2 enzyme do? It will cause two cuts in the DNA strand. Okay, and because of the two cuts, again the DNA will uncoil itself. But later on, after the two DNAs are formed, it has to again coil. So again, it will join the two cuts which it had done in the initial part, and again you can see the DNA, the two DNAs will be formed, which will be helically shaped in nature. So you have topoisomerase 1, which causes one cut in the DNA strand, and topoisomerase 2, as the name suggests, it will create two cuts in the DNA strand. So double cut in the DNA strands. Okay. Now, what is the role of topoisomerase inhibitors? The chemotherapeutic agents which act on the topoisomerase enzyme, and they inhibit the topoisomerase 1 or topoisomerase 2 enzyme, all are called as topoisomerase inhibitors. So what is a topoisomerase inhibitor? Topoisomerase inhibitor is a chemotherapeutic agent which inhibits topoisomerase 1 enzyme, or it can inhibit topoisomerase 2 enzyme. Okay, so if it inhibits topoisomerase 1 and 2 enzymes, what will happen? The DNA cuts cannot take place. If the DNA cuts do not take place, then the uncoiling of the DNA will be stopped. If the DNA cannot uncoil, then obviously DNA synthesis cannot take place. So that is the role of topoisomerase 1 and topoisomerase 2 enzyme. You have different types of drugs which actually act. Yeah, topoisomerase 1 inhibitors are irinotecan and topotecan, which we will be doing. And topoisomerase 2 enzymes are, topoisomerase 2 inhibitors are etoposide, and the drug's name is Actitop, which we have. We will be doing these two products when we actually do the products. So you have to understand that irinotecan and topotecan are topoisomerase 1 inhibitors, and etoposide is a topoisomerase 2 inhibitor. Okay, and topoisomerase 1 inhibitor will not allow one cut in the DNA strand. Topoisomerase 2 inhibitor will not allow the two cuts to take place in the DNA strand, because of which the DNA cannot replicate itself, and therein the further cell multiplication is stopped. Okay. Sorry. Yeah. Now, first we saw the antimetabolites, then we saw topoisomerase inhibitors. Now we will be seeing chemotherapeutic agents which act on the M phase. M is the actual phase where the cell division is taking place, or it is a cell mitosis phase. Yeah. Now, when I was explaining you DNA synthesis, we had seen the DNA actually duplicates itself. Okay, when the DNA duplicates itself, what happens is mitotic spindles. You can see these mitotic spindles, right? These mitotic spindles are formed, and on one side they are attached to centromeres. These are centromeres. This is a centromere. This is a centromere. So sorry, centrosomes. So this is a centrosome. This is a centrosome. From centrosome, the mitotic spindles are getting released here, 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 here. Now these mitotic spindles will go and attach themselves on the DNA to a place called the kinetochore. So where they are getting attached is called as a kinetochore in the DNA, right? Now, how these mitotic spindles are formed? These mitotic spindles are formed with the help of alpha tubulin and beta tubulin. Okay. Now, what is basically formation of mitotic spindles? This is basically called as polymerization. So, in polymerization with the help of alpha and beta tubulin, you have the formation of mitotic spindles. So they keep increasing. So that is called as polymerization. They keep increasing because they are required to pull the genetic material. So first these mitotic spindles have to polymerize, grow, and attach themselves to the DNA strand. Okay, so it this. But later on, what happens after they have gone and attached to the DNA molecule, the DNA, one pair of chromosome has to be brought to one side and the other pair of chromosome has to be brought to the other side. Okay, so how that will be pulled? When you release the tension in the mitotic spindles here. Okay, so when you release the tension, what happens is they come back. Okay, so what is that procedure called as? It is called as depolymerization. When the tension is released, okay, then the mitotic spindles depolymerize, and then the genetic matter or the chromosomes are pulled close to the centrosomes. Okay, so this procedure where the mitotic spindles tension is released is called as depolymerization, right? So why we are studying this? Because there are drugs which are going to act on these mitotic spindles, right? Now, uh, basically, this mitotic spindles have to be formed, so you have polymerization. But if the mitotic spindles are not allowed to be formed, then obviously the genetic matter cannot be pulled back. So, which are those drugs which do that? Vinca alkaloids are the drugs which actually, of course, we have given a name called Vinca alkaloid. Vinca alkaloid are alkaloids which are derived from plants, and they are derived from plants called as Vinca. They are called as periwinkle plants. So the products which are taken from periwinkle plants are called as Vinca alkaloids. Okay, and these Vinca alkaloids inhibit polymerization. So what do you mean by that? They do not allow the mitotic spindles to be formed. They will go and attach themselves here, because of which these mitotic spindles cannot be formed, and due to which what happens is the DNA strands, the mitotic spindles will not be formed and they cannot attach themselves to the DNA. Okay, so this is how the Vinca alkaloids act. Now, which are the molecules present under Vinca alkaloids? You have vincristine, you have vinblastine, you have vinorelbine. So it is inhibiting the polymerization or the formation of the mitotic spindles. Okay, and the molecules which are classified under these are called as vincristine, vinblastine, vinorelbine. Okay, now there are drugs which inhibit the depolymerization. Now, what do you mean by that? First, polymerization has already taken place. That means the mitotic spindles have already formed. Okay, and they have gone and attached to the DNA strand here. But they have to be pulled back so that they come close to the centrosomes and two daughter cells can be formed. Okay, so that procedure where the mitotic spindle's tension is released is called as depolymerization. But there are a class of drugs called taxanes which do not allow the depolymerization to take place. Why? They will go and attach themselves to these mitotic spindles. Okay, if they go and attach to these mitotic spindles, then an assembly is formed. Okay, and that cannot destabilize. They are going and attaching themselves to the mitotic spindles and they make the mitotic spindle static. They cannot move, they cannot depolymerize. So what happens if they cannot depolymerize? They will not pull back the DNA strands. Okay, so the molecules which come under these category are called as paclitaxel, docetaxel, cabazitaxel, and all of them are classified under taxanes. Okay, and the role of the taxane is to inhibit depolymerization. It will not allow the depolymerization to take place, because of which the mitotic spindles cannot pull back the DNA to the two daughter cells, and if they cannot do that, then obviously cell mitosis cannot take place, and this is happening in the M phase, which is the mitotic phase. Okay, I hope this is clear. You have microtubule inhibitors. Why they are classified as microtubule inhibitors? Because they are inhibiting the microtubules which are required in the M phase for cell mitosis. Right? One class of drugs inhibits polymerization. They are called as Vinca alkaloids. The other class of drug inhibits the depolymerization, and those class of drugs are called as taxanes. In today's scenario, if you see, taxanes are very, very widely used in treating cancer, and the most widely used molecule in taxanes is paclitaxel. It is one of the most widely prescribed or the heavily prescribed molecule worldwide, which is paclitaxel, and after which came docetaxel, and today you also have cabazitaxel, but still the usage of paclitaxel is very, very high. I hope this microtubule inhibitor is clear. Okay, when we go to the products, again there'll be a revision. But if you understand microtubule inhibitors, then understanding the products will be very, very easy. Okay. Now we are coming to another class of drug called as anti-tumor antibiotics. Basically from the name itself, you can make out that they are a class of antibiotics. Okay, but they are not your regular antibiotics. These anti-tumor antibiotics are also called as anthracyclines, and they are derived from a microorganism called Streptomyces bacteria. Therefore, they have been given a name called antibiotic, but their action is anti-tumor in nature. Therefore, it is anti-tumor antibiotic. But in anti-tumor antibiotics also you have two class. One set of anti-tumor antibiotics is bleomycin, and the others are doxorubicin, daunorubicin, epirubicin, and idarubicin. If you see any chemotherapeutic agent with where their name is ending with CIN, then they are classified as anti-tumor antibiotics. That is something that you have to remember. And they were originally derived from a microorganism called Streptomyces bacteria, and they are also called as anthracyclines. Now, what is the mode of action, or how the anti-tumor antibiotics basically act? They are basically acting in the G2 phase, where the cell is preparing itself for the cell mitosis to take place. Now, when bleomycin is given, what happens? Bleomycin actually binds to the DNA and produces highly reactive free radicals, which leads to cessation of DNA strand. So, what do you mean by this? Basically, bleomycin will go and attach itself to the DNA strand and will release highly reactive free radicals. Now, what do you mean by free radicals? See, when any chemical is given, say for example, carbon has got its valency as 4. So it will go and attach to 4 different hydrogen atoms or 4 different atoms and complete its valency, right? But if you remove one hydrogen atom, or if you remove 2 hydrogen atoms, then it becomes a free radical. So what do you mean by free radicals? It can attach 1 or 2 more atoms to complete its valency, right? But till the time it is not attaching, it remains a free radical. So it can go and attach anywhere, right? So that is basically a free radical. Now, when DNA is getting attached to bleomycin, these free radicals are formed. So these free radicals can go and attach themselves anywhere on the DNA, and that leads to the cessation of DNA strand. That means the DNA strand gets damaged, and because of which, if the DNA strand gets damaged, the DNA replication will not take place and cell mitosis does not take place. So because of this free radical which is generated, the DNA gets damaged. Now, what happens to molecules like doxorubicin, dono, epi, and idarubicin? Of course, these anthracyclines also undergo chemical reactions to form highly reactive free oxygen radicals. There you had free radicals, it could be any free radical, but here you have oxygen free radical. Oxygen's valency is 2, so 2 oxygen atom attach itself to form a oxygen molecule. Okay, but if a free oxygen molecule is there, it becomes a oxygen radical. Now, the free oxygen radical can damage the cell membrane. The cell membrane, that means not the nuclear membrane, the outer membrane of the cell. It can damage the cell membrane, it can damage the proteins, it can damage the lipids, and they also trigger apoptosis. What do you mean by apoptosis? Basically, it can damage, basically, it is called as programmed cell death. Okay, it can lead to the death of the cell. So, that is programmed cell death. So, basically, these anthracyclines like doxorubicin, daunorubicin, epirubicin, and idarubicin, they generate free radicals. Okay, but which type of free radicals? They generate free oxygen free radicals, and they are not attaching themselves to the DNA. They are going and attaching themselves to the cell membrane, and they internally they damage the cell membrane, and they can damage the proteins, the lipids, and this can actually lead to cell apoptosis, that means programmed cell death. Okay, so this is the simple mode of action of anthracyclines. Bleomycin forms free radical, but they are not oxygen free radicals. They are highly reactive free radicals of other elements, and they are binding to the DNA. Whereas the other anthracyclines like doxo, dono, epi, and idarubicin, they of course create free oxygen radicals which damage the cell membrane. It can damage the protein, it can damage the lipids, and because of which it can lead to cell apoptosis. So this is a simple mode of action of anti-tumor antibiotics. They are also cell cycle specific.
Again, you have muted, ma'am. You have muted.
Yeah. Then you have another class of drug which is cell cycle non-specific. They are called as alkylating. Of course, at the moment, you don't have any drugs.
Ma'am, you have muted again.
You know how this is happening. Yeah. Yeah. Basically, when you have any alkanes, if one hydrogen atom is removed or two hydrogen atoms are removed, then it can give you an alkyl group. Okay. So these alkyl groups have got a free valency, so they can attach themselves to any other molecule or any other element for that matter to complete its valency. So these alkylating agents are chemotherapeutic drugs who have got one or two alkyl groups in it. Therefore, these alkyl groups have the nature of going and binding itself somewhere. So these alkylating agents, when they have alkylating groups in it, they can go and bind themselves to the guanine base. So here you can see the skeleton. Yeah. So this is the alkyl group. So this is getting and this is going and attaching itself to the guanine base. Okay. So when it goes and attaches itself to the guanine base, you can see cross linkage between the functional groups. Okay, which in turn will prevent the DNA to separate, and therefore their DNA synthesis will not take place. So this is the basic simple mode of action of alkyl group. They go and bind themselves to the guanine base because they have a free radical. If they go and bind themselves to the guanine bases, they can cause cross linkages, which in turn will prevent the DNA to separate. Because see, this is going and attaching themselves to the guanine base. It can cause cross linkages. Okay, this and this is going and attaching itself. Here also it is going and attaching itself. So what will happen when these alkyl groups have attached themselves to the guanine base, the two DNA strands which you can see here cannot separate. Okay, if they cannot separate, then obviously DNA cannot uncoil and further I mean, the DNA duplication will not take place. If DNA duplication will not take place, then obviously cell cannot multiply itself. So basically, these chemotherapeutic agents have got alkyl groups in it. These alkyl groups can bind themselves anywhere, but in alkylating agents, what happens? These alkyl groups go and pair with the guanine bases, and when they go and pair with the guanine bases, they lead to cross linkages. When this leads to cross linkages, and obviously DNA is prevented from separating. When DNA cannot separate, then obviously DNA synthesis or DNA replication cannot take place. You have, of course, it prevents the DNA synthesis. And there is one more way in which what happens is even the guanine base pairs can pair themselves wrongly. If the guanine base pairs pair themselves wrongly also, then obviously DNA synthesis cannot take place. The right pairing has to take place. The guanine can go and pair itself with adenine or with thymine, whereas guanine has to pair itself with cytosine. If it does not go and pair itself with cytosine, then obviously right base pairing cannot take place, right? So this is a simple mode of action of chemotherapeutic drugs. We have multiple drugs which are coming under alkylating agents like nitrogen mustards. You have chlorambucil, cyclophosphamide, ifosfamide, melphalan. Then in nitrosoureas, you've got carmustine, lomustine, simustine. In triazines, you have dacarbazine, temozolomide. Of course, we have some of the drugs here, but in Ethiopia, you are not promoting those drugs like temozolomide and cyclophosphamide. You are not promoting. So we will not be discussing any of these molecules, right? So this is the way how these alkylating agents basically act. So we have the platinum compounds also. Currently, you don't have any of the platinum compounds in Ethiopia, like cisplatin, carboplatin, sorry, you have got oxaliplatin is there, right, right, right. So these chemotherapeutic agents, these are cell cycle non-specific. They have platinum group in it. When these platinum compounds come in contact with the H2O, that is water molecule, they release hydroxide molecule. Now, these hydroxide molecule can form intrastrand adducts. They can go and attach to two bases and can call intrastrand adducts, and they can also go and attach to base pairs in two different helicals and can cause interstrand cross linkages because of this platinum compound which gets hydrolyzed. After hydrolysis, they can attach themselves to two base pairs in the same helix, or they can attach themselves in two different helix, therefore causing intrastrand adducts. That means they form intrastrand adducts between the strands, and between two different strands, if they go and bind themselves, it is called as interstrand cross linkages. But you have this intrastrand adducts or interstrand cross linkages being formed. What happens? The DNA strand cannot uncoil because an adduct is formed. If they cannot uncoil, then further DNA synthesis cannot take place, and obviously cell cannot undergo cell mitosis. So this is a simple mode of action of platinum coordination compounds, and they are cell cycle non-specific. That means they can act on all the phases of the cell cycle, either the G1, G2, M, or the G0. You've got cisplatin, carboplatin, oxaliplatin. You also have the resulting cross-linkage inhibits DNA repair and DNA synthesis, which we have already. Okay. Of course, these are all the chemotherapeutic agents that we saw, but there are certain hormonal therapies also which are used in treating cancer, and we also have some of those molecules with us. So therefore, I will touch on the hormonal therapies also. So what is the role of hormonal therapy in treating cancer? Hormone therapy slow or stop the growth of hormone sensitive tumors which require certain hormones to grow. I'm sure you might be knowing there are various types of cancers like breast cancer, prostate cancer, which grow in the presence of the respective hormones. Like breast cancer grows in the presence of estrogen. If it is ERPR positive, then it is estrogen and progesterone. When you talk about prostate cancer, then the testosterone levels are very high. So these hormones are responsible for these type of cancers. So if these hormones are present in large quantities, then these cancers can multiply freely. Okay, therefore, what is required? You have to stop the production of these hormones, or you have to inhibit these hormones, and this is how you will control the cancers which are hormone dependent. Okay, like breast cancer, prostate cancer. I have. Of course, you don't have to go into much of detail. We will be doing this. As I told you, hormones used in breast cancer, hormonal therapies which are Getting some disturbance. Yeah, there are different types of hormonal therapies which are used for treating breast cancer, which we will be doing in detail. Like you have aromatase inhibitors, which will actually, from the name itself, you can make out they will go and inhibit the aromatase enzyme, which is actually responsible for converting androgens into estrogen. And you have molecules like anastrozole, exemestane, letrozole, which come under the category of aromatase inhibitors. You have selective estrogen receptor modulators like tamoxifen and raloxifene. We will be explaining you tamoxifen. Then you have estrogen receptor antagonists like fulvestrant and tormephin, which of course we don't have. Then you have luteinizing hormone releasing hormone agonists such as goserelin, leuprolide, and triptorelin. Of course, we don't have any molecule. Now, like chemotherapeutic drugs have their side effects, which I will be explaining you. The hormonal therapies also have some of the side effects which we know when patients take hormonal therapy, especially when females undergo some hormonal therapy, there can be fluid retention. Because of fluid retention, the patient's weight gain is also seen, and there is swelling and edema. When patients take hormonal therapies, females, of course, get hot flushes also. There can be interrupted menstrual periods depending upon the type of hormonal therapy which are given. Patients can become impotent also with some of the hormonal therapy. So these are some of the side effects of hormonal therapy. Okay, so we have gone through chemotherapeutic agents. We just saw some of the hormonal therapies. Of course, you have various other classifications also, like targeted therapy and various bone marrow transplants and such things. But we will not go into that because we don't have any drugs falling under those categories. But there are certain terminologies which I want you all to understand. So I will quickly take you through that. Uh, Basically, these terms will be used when we talk about the different indications of the molecule. So one is induction therapy. From the name itself, you can make out induction means a treatment which is given in the beginning, at the first in a series of therapeutic measures taken to treat a disease, typically a cancer. The induction therapy, for example, in acute leukemia is the initial chemotherapy designed to bring about a remission. As the name suggests, induction therapy is basically a treatment which is given for the first time to the patients. So it's the induction therapy. Just one second. Yeah, sorry. When a patient is coming for the first time and the doctor decides a treatment which has to be given, any chemotherapy that has to be given, that becomes the induction therapy for the patient. So the chemotherapy that is given for the first time to a patient is called as induction therapy. Okay, irrespective of the stage, whether it is the metastatic stage or the stage 1, stage 2, stage 3, any stage. So the chemotherapy that you give to the patient for the first time is called as induction therapy. Then what is first line treatment? First line treatment is basically the chemotherapy that you give to the patient for the first time, but the patient's disease is in the metastatic stage. Okay, what do you mean by metastatic stage? When the patient's primary tumor has gone and spread to some other organ. Okay, that means secondaries have developed. It has gone and metastasized. It has gone and spread itself to another part. Okay, that is metastasis. So the chemotherapy you give to cancer patients whose disease has metastasized is the first line treatment. The first chemotherapy that you're giving to a patient whose disease is already metastasized is called as first line treatment. Now, what is second line treatment? After the first line treatment, the patient has shown some improvement or has shown some improvement for some time, but again the disease comes back after some time, or the first line treatment which was given has not suited the patient. The patient had some severe toxic effects. Okay, so the patient has to move to another treatment or has to move to another chemotherapy. So that is a second line treatment. So the chemotherapy which you give for the second time to a patient whose disease is already in the metastatic stage is called as second line treatment. Similarly, you have third line treatment also. If the patient's first two treatments have failed or have not shown the desired result, then the patient has to move to another chemotherapeutic agent or a combination of chemotherapy, then that becomes a third line. Okay, so this is how you have to understand. Now, what is maintenance therapy? When you give chemotherapy to the patient whose disease is in the metastatic stage, that is a first line treatment. Then you have a set protocol in the second line treatment that the patient will get this. But when you have to increase the gap between the first line and the second line treatment, then you give some chemotherapy, generally a oral type of a chemotherapy, to prolong the duration between first line and second line. So that is called as maintenance therapy. Okay, so the treatment which is given to the patient between first line and second line to prolong the duration between the first line and second line treatment is called as maintenance therapy. So I have just given you these terms so that you understand the indications in a better way. So what is induction therapy? The chemotherapy that is given to the patient for the first time. Okay. And what is first line? The chemotherapy you give to the patient when the disease has metastasized. The chemotherapy which you give to the patient for the second time is called a second line. Similarly, third time is a third line treatment. Maintenance therapy is the therapy which is given between first line and second line to prolong the duration of the two treatments or to increase the gap between the first line and the second line treatment. The more the gap, the better it is for the patient, right? So these are some of the therapies and the terminologies that you have to remember and know. Now, we have been discussing about cancer. I'm sure you know this, but just to give you an idea, what do you mean by medical oncologists, radiation and surgical oncologists? These are the different types of oncologists which we meet in our regular practice. But what do you mean by medical oncologist? Medical oncologist is a doctor who treats cancer with only drugs, like the chemotherapeutic drugs. Medical oncologist can see solid tumors also, and he can see even hematological malignancies also. But he's treating the patient with chemotherapeutic agents only. Okay. Then you have radiation oncologist who treats cancer with the help of radiations, and sometimes he might give some chemotherapy like concurrent therapy also along with radiation therapy. Okay, then you have surgical oncologist. A surgical oncologist is a surgeon who specializes and removes the biopsy samples. That means he removes a part of the tumor which can go to the pathology lab for biopsy. He also is a surgeon who removes the cancerous tumor or the cancerous growth and also the nearby tissues. Is any disturbance? I can continue. Yeah, Kimia, I can continue.
Yes, please.
Okay, okay. So the surgical oncologist helps in sending the sample for biopsy. He also resects the cancerous growth by removing the malignant tumor, and he also basically removes. Just 1 second, just 1 second. Okay. Yeah, I'll start. So we just saw what is the role of a medical oncologist, what is the role of a radiation oncologist. So now we'll understand what is the role of a surgical oncologist. Can anyone tell me what is the role of a medical oncologist?
Sorry.
What is the role of a medical oncologist?
Here in Ethiopia,
What is the role of a medical oncologist?
as you mentioned, is a specialist doctor who treats the cancer patients with the drug,
Yeah.
the chemotherapy. Yeah.
Yeah. So basically, which type of drugs?
Chemotherapy drugs,
Yes. And he treats which type of cancers?
any cancers.
Any cancer?
Yeah.
Solid tumors, and they are specifically called as solid tumors. And
Sorry.
Kimia, you can ask the others also to reply.
Okay.
Solid tumors, and which are the other types of cancers which I told you?
ከነገርኳችሁ ውስጥ ቲመር ካንሰሮች አሉ፣ አደር ካንሰሮች አሉ። የትኛውን ካንሰር ነው ጥያቄዋ?
Medical oncologist treats which type of cancers? Solid tumors and
Solid tumors and what? Okay, well,
no one?
they said tumor cancers also, if the stage is serious in the surgical.
What?
I do that. Yes.
Anyone wants to explain?
ምን ተናገረች? በነገራችን ላይ ቲመር ሜዲካል ኦንኮሎጂስት ቲመር ካንሰር አንድ ትሪት ያደርጋል አለች አይደለ? እሷ ራሱ መለሰችው። ሌላ ምን አይነት ታይፕ ኦፍ ካንሰር ትሪት ያደርጋል? ብሬስት ካንሰር።
I hope all of you are meeting the medical oncologist there.
Here in Ethiopia, medical oncologists are treats like breast CA, like tumors, CA, cancer.
Yeah, correct. So
And
these are all
then-
solid. These are all solid tumors, right? So CA breast is solid tumor, CA lung is solid tumor. Are they also not treating some other type of cancers like hematological malignancies?
Hematologists usually do that.
No, see. A medical oncologist can treat solid tumors with chemotherapeutic agents. They can also treat hematological malignancies like all types of leukemia, all types of lymphoma. Okay. All types of leukemias. There are various types of leukemias, which of course I will tell you: CML, ALL, they are called as chronic myeloid leukemia, lymphomas like Hodgkin's lymphoma, non-Hodgkin's lymphoma, also multiple myeloma. So all these are hematological malignancies. So medical oncologists can treat solid tumors and all types of hematological malignancies, right?
Here in
With
Ethiopia
the help,
also,
yeah.
Here in Ethiopia, medical oncologist has a subspecialty of hematologist. So usually they are treating these malignancies like leukemia, blood cancer, and other.
Yeah, see, you're correct. Medical oncologist, and you have hemato-oncologist who treats all these hematological malignancies. So that means in Ethiopia, the medical oncologist is not treating at all the hematological malignancies, right?
Yeah.
Okay, so in that case, medical oncologist for you there is only treating solid tumors with the help of chemotherapeutic drugs, right? Radiation oncologist treats cancer with radiation therapy. Surgical oncologist is the one who treats cancer by removing the cancerous growth, by removing the surrounding tissues which are near the cancerous growth, and he also does the biopsies. He removes the tumor, and the sample of the tumor can be sent for biopsies. Biopsy is basically the confirmatory test for cancer, right? So a surgical oncologist is doing this.
Yes, you're right.
Yeah. As you rightly mentioned, you have hemato-oncologist who basically treats all the hematological malignancies, that means blood-related diseases and also diseases related to the lymphatic system, right? So he is the one who treats the blood-related diseases, blood-related cancers, and also all lymphatic system-related cancers. He treats with chemotherapy, and he can also be doing bone marrow transplants. Okay. So these are the four specialties that we deal with: medical oncologist, surgical oncologist, and hemato-oncologist. Then there are also pathologists who examine the tissues of the tumor samples which we send. Like for biopsy, you sent a sample of the tumor to a pathology lab, and there is a pathologist who actually examines it under the microscope and sees whether it is a cancerous growth or not, or what is the grade of the tumor. And it basically, the grade of the tumor helps in finding out whether the tumor is fast multiplying or slow multiplying. Okay, so you have the pathologist also who helps in the diagnosis of the cancer. So basically, we'll be dealing with all these specialties. So can anyone tell me there are how many specialties of cancer doctors in oncology?
54.
Yeah. Can someone name for me?
Sir?
Can you name them for me?
Sorry, it's not clear. Again, please.
Can you name all the specialties of
The doc-
oncologists? I can't hear you all.
Yes. ስፔሻሊስት ነው የፈለገችው። ስሙ እሺ By the way, ቀላል ይመስላል ግን መልሱላችሁ ለተሶች ማለት ማይነር ነው ላችሁ ስማትበሉ ማለት ነው።
I can't hear.
They are mentioning all like oncologist, hematologist, pathologist, surgical oncologist.
List them properly to me. What do you mean by oncologist? All four of them are oncologists.
Medical oncologist. They said medical oncologist.
Yeah, they're called as medical oncologists. Then,
Yes.
which else? Hematologist. Go in the order. Medical oncologist, then
Yeah, radiation oncologist.
radiation oncologist, correct.
Surgical oncologist.
Correct.
Hemato-oncologist.
Yes, right. So you have 4 types of oncologists with whom we deal with, right? And all 4 of them have the capacity of prescribing chemotherapeutic drugs depending upon the institute they are present in and depending upon the condition that they are treating, right? So all 4 of them are our customers. Okay. So this is something that we have to remember because we are going to be meeting oncologists day in and day out, right? So can a surgical oncologist treat solid tumors?
Who?
Can a surgical oncologist treat solid tumors?
Solid tumors?
Yeah.
Yeah, solid tumors will be treated by medical oncologists.
By medical oncologist. But medical oncologists will treat with the help of
Surgical oncologist, radiologist, radio-oncologist.
No. What was my question? Can a surgical oncologist also treat solid tumors? What do you mean by solid tumors? All the cancer, which is all the tumor other than your hematological malignancies like leukemia, lymphoma, multiple myeloma, all the others, all the liquid types are coming under hematological malignancies, and all the remaining are solid tumors. So who can treat solid tumor?
Medical oncologist, surgical oncologist.
Yes. How will a surgical oncologist treat? Even a surgical oncologist can do a surgery and remove the solid mass of the cancer, right? So a surgical oncologist can also treat a solid tumor case. Medical oncologist anyway can treat both. Hematological malignancy can be treated by hemato-oncologist. Radiation oncologist can also treat solid tumors, hematological malignancies also, but with radiations, right? So you need to understand all of them can do this, but their specific roles are there, right? So is it clear till now?
Yes, clear.
You have any doubt?
No.
We can proceed?
Yes, please.
Okay. So we saw chemotherapeutic drugs, we saw their classification, we saw their mode of action, we have seen chemotherapeutic agents' role and their side effect. Okay, so and now we will be seeing the side effects. So we need to understand the side effects because each chemotherapeutic agent has got different side effects, but I will be touching upon the major side effects. So some of the major side effects of chemotherapy, the first and the biggest one is myelosuppression. As I told you, chemotherapy I mean, basically, chemotherapeutic agents act on the fast multiplying cells. The fast multiplying cells are in the bone marrow because you have production of blood cells continuously there. You have the RBCs, you have the WBCs, you have the platelets being formed. Okay, so when chemotherapeutic agents are given, they will affect the bone marrow. So if there is depletion in the bone marrow, if there is depletion in the cells in the bone marrow, that condition is called myelosuppression. But those conditions can be different. If you have low WBC counts, then that condition is called as leukopenia. If you have low platelet counts, then they are called as thrombocytopenia. If the RBC levels goes down, then that condition is called anemia. And all these side effects can be managed by different drugs. You know, anemia, you can give iron supplements. For treating leukopenia, you give filgrastim like granular stimulating factor GCSF. Filgrastim is given to treat leukopenia. So there are different drugs and different molecules which can be given to treat these side effects which are associated with the chemotherapeutic agent. The other common side effects, which are the GI side effects. As I told you, the GI lining has got mucosal lining and of course salivary glands in the mouth. So the salivary glands can be affected, the mucosal layer can be affected, because of which you have the GI side effects like nausea, vomiting. Okay, patients frequently have the urge of vomiting. So that condition where they feel like vomiting is called as nausea. And then you have vomiting as one of the side effects. It can be acute or delayed. That means it can be sudden or it can take some time. It can also cause dehydration, and of course, patients have to be given antiemetics. You have many antiemetic molecules like palonosetron, granisetron, aprepitant, which is being used, and you have combination of antiemetics also being used for treating GI related side effects like nausea and you don't have any drug in the antiemetic range, but there are multiple combinations which can be given. Then there is another side effect called loss of appetite, and it is also called as anorexia, which is very commonly seen in cancer patients. There are other GI side effects like gastritis and mucositis, which are associated with the chemotherapeutic agents. Then one of the biggest side effects, as I told you, is alopecia, which is hair loss, which is of course temporary. Till the time patient is taking chemotherapy, the patient loses hair, but after some time, the patients get back their hairs, and of course, it can cause temporary discomfort to the patient also. There are other side effects which are associated with chemotherapy, like mutagenicity, carcinogenicity. Mutagenicity is basically it induces genetic mutations in the DNA. Carcinogenicity is basically these chemotherapeutic agents can themselves become like carcinogens and cause different types of tumors. Patients can lead to infertility where a female or a male cannot conceive. Then nephrotoxicity is basically toxicity in the nephrons and the kidney. Cardiotoxicity associated with the toxicity which is seen in the heart. Neurotoxicity which is seen in the neurons, which is in the basically nervous system. So these are all the side effects which are associated with chemotherapy, which can be which can be temporary or it can be permanent also. So these are the most commonly known side effects of chemotherapeutic agents. There will be some specific which are associated with the drugs, but when we go to the respective chemotherapeutic agent, we can take that in detail. So this was all that I wanted to take with respect to uh, basics of oncology, though the subject is very big, but I have restricted it to this much because this much will be sufficient for you to understand the product. So if you all have any doubts, then please free to ask me. Anyone? Kimia, if anyone has got any doubt, they can get it cleared, because if this is clear, then understanding of the product will be very, very easy and simple.
Yes, I am asking them, but all of them say it's clear.
It's clear? Really?
Yes.
Okay, okay. So you want to break for lunch for 1 hour?
Yes, for you. Yes, lunch for you and refreshment for us.
Yeah, okay, okay. Refreshment for you all. Okay, so we'll break for 1
Yes.
hour. Exactly 1 hour. Here it is 1:20, so we'll start at 2:20 sharp. Yeah, exactly 1
Yeah,
hour
nice.
from now. Yeah. Okay.
Okay,
Shiva,
good.
you can stop the recording now. Please come back after 1 hour. Yep. Yeah. Shiva,
Thank
you're
you
there?
very much. Okay.
Okay, 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 okay.